Hi everyone! Today I'm going to be talking about digits and fin rays sharing a common developmental history, and then on Tuesday, Katie's going to take over for the discussion. So I wanted to talk about how and why these researchers decided to do this study. So they wanted to better understand the evolutionary transformation of fish fins to tetrapod limbs, and they used CRISPR and Cas9 as well as fate mapping of 5' Hox genes and enhancers that are necessary for the development of wrists and digits in tetrapods in order to do so. Just a side note, fate mapping is basically tagging one or more cells, and as those cells proliferate and migrate, you can see where they end up. So just a little bit of background. Fin distal skeleton have those characteristic fin rays, which are made up of dermal bone tissue. And throughout profound changes throughout the course of evolution, we have the tetrapod limb, which have characteristic digits made of endochondral bone tissue. And we know that these profound changes are the decreasing of fin rays and the expansion of distal endochondral appendage bones, but how exactly this occurred is still unknown. So the authors wanted to begin by looking at how digits arose in the first place. So from previous knowledge, we know that Hox A and Hox D enhancers of late and early phase expressions gives rise to proximal and distal segments, respectively. So they showed this through those knockout experiments of those Hox genes in mice, and that resulted in the lacking of wrists and digits exclusively. But this mechanism has not really been explored in fish fins, so it's unclear whether there are cellular and genetic markers of the autopod that reside in fish fins. In order to do so, they marked cells, Hox enhancers, and then followed the fates of those cells. They also performed knockout experiments of Hox genes, specifically Hox A13A, Hox A13B, and Hox D13A. And they also used in situ hybridization of Hox genes to determine if active Hox expression has a role in pectoral fin fold development. So what did they see? Looking at the top, we see Hox A13A expression, and on the bottom, Hox A13B expression. So both are expressed in the distal fin mesenchyme at 48 hours post-fertilization. And as we move from that 72 to 96 hour mark, it's becoming more weakly expressed in the proximal portion of the pectoral fin. And then at 120 hours, there's very weak to no expression seen. So then this is looking at Hox D13A, and we can see that it is expressed in the posterior half of the fin, but it does weaken after 96 hours post-fertilization, and after 10 days, there's no expression at all. This led them to the conclusion that Hox13 genes don't play a main role in the fin fold development past 72 to 96 hours post-fertilization. So this led them to their next experiment, which was to determine which structures Hox-positive cells populate in developing adult fin folds. They did so by looking at enhancer expression. CNS65 was used to follow the fate of cells that have early phase activity, and E16 was used to follow cells with late phase activity. E16 is a gar Hox A enhancer taken from the spotted gar, which is a ray-finned fish that's more ancient than the zebrafish, so it may have some evolutionary significance in terms of its genome from the past. And in previous experiments, this enhancer was shown to be a marker for hand and digit formation by doing a swap. So when you put the enhancer in mice, you get normal digit and wrist formation. So what did they see? They were looking to see where early and late phase enhancer drives expression in this experiment. So on the top, we're looking at early phase through that CNS65, and on the bottom, late phase through the E16. So focusing on the top row first, CNS65 enhancer is expressed through the fin fold from 31 to 38 hours post-fertilization, whereas late phase expression driven by E16 begins somewhat weakly in the distal fin at 38 hours post-fertilization and then dampens around that 72-hour mark. So it seems that E16 is driving more distal expression, which is consistent for something that is making digits. So this is the cell staining with M. cherry to see where these cells with the Hox enhancers end up. So in 2B, we're seeing cells with that CNS65 enhancer. So mesenchymal cells, where that enhancer is expressed, make up the endochondral disc of the pectoral fin by 20 days post-fertilization. So that's contributing to the fin fold and the endochondral disc, which suggests that the cells are migrating distally outward of the endochondral disc. And then looking at the late phase expression, again in red, we're seeing that at the 20-day mark, the red staining has continued to stay more towards the tip of the fin bud. So the cells that are undergoing that late phase expression are present mostly in the fin fold, though some are in the distal edge of the disc. But this indicates that cells are actively moving out of the fin fold, and that late phase Hox positive cells are likely to migrate from the endochondral portion of the fin into the fin fold. Here we are seeing the fin of an adult zebrafish. So those red staining is that M. cherry with the late phase Hox expression overlaid with an osteoblast marker, and so osteoblasts make up fin rays. So we can see that the late phase Hox expression is really only present in what was used to be the fin fold, and not in the endochondral disc at all. Since the E16 enhancer is only present in the distal endochondral disc, and the labeled cells end up in the fin rays of the adult, late phase Hox positive cells are likely to migrate from the endochondral portion of the fin into the fin fold, so that's migrating out. So the researchers then wanted to determine the function of Hox13 genes. And they did this by performing knockout experiments on Hox13 genes in the zebrafish using CRISPR-Cas9. So we get three views of the fin ray. In the top, we're seeing the wild type, and then we see the knockout of the HOXD13A, which resulted in no phenotypical change. When they knocked out HOXA13B, they saw shorter fin rays, and that was the same case when they knocked out HOXA13A. When they knocked out both of those genes, they saw a greatly reduced fin ray, and when they performed a triple knockout, they saw a greatly reduced fin ray and altered proximal radials. This led them to the conclusion that the late Hox genes are involved with the patterning and proximal endochondral radials of fin formation. So coming back to those shared developmental histories, even though the skeletal tissues that make up these structures are different, there are similar late phase expression of those Hox genes driven by a shared regulatory structure. So that kind of points to a developmental homology, and they show that through the loss of 5' Hox activation. So just tying this all together, previous research shows that mice, when they have the normal expression of late phase Hox enhancers in that red portion of the limb, you get the normal wrist and digit formation. And then when you have a double knockout experiment, you lack those structures exclusively. And so similarly in the zebrafish, there's this mid zone of late Hox expression, and then when those cells migrate outwards, you get the normal development of those fin rays. But if you have a double knockout again, you have an increase in endochondral radials and a decrease in those fin rays. So that's a really important step to this transition. This led the researchers to a future hypothesis. So we know that this is a result of the deficient migration of mesenchymal cells. So how exactly that leads to the shortening of fin rays and increase of endochondral radials is something that should be looked at. Some major trends that we can see is the elaboration of the endochondral bones, as well as the progressive loss of the extensive dermal fin skeleton, and this may be linked at the cellular and genetic level. So this study was significant because it revealed a cellular and genetic connection between fin rays and the digits of tetrapods through the transition of cellular fates. Thank you for listening, and I look forward to our discussion on Tuesday.